As I said at the beginning, we have three very different approaches to capturing solar energy and either generating electricity or a fuel. And in this talk, you'll see the biochemical perspective. Clint Chapple from Purdue University will present his work that's funded by GCEP.
All right, well, it's a pleasure to be here, and I'm grateful for the opportunity to present our work that is funded both by GCEP and also from funding through the Department of Energy. I think I could have retitled my talk now for something completely different, because it's going to be a big departure from the talks we've heard so far. And that's not working. Okay. I'm probably pushing the wrong button. Or my other right. It's okay. So I'd like to give you a brief outline of what I want to talk about today. Again, this talk and our research is focused on lignin, the hardening component of the plant cell wall. What I often boil it down to is the difference between a piece of celery and a piece of wood is the presence of lignin in that cell wall. And you know what implications that has for the durability of a piece of wood, whereas a piece of celery is entirely and rapidly compostable. However, that polymer is very important to the plant, and I'll give you a short overview of that. I want to tell you why GCEP and other funding agencies have focused funding on lignin, because of its plasticity. And I'm also going to tell you a little bit about what happens when we mess around with it in ways that the plant doesn't like, because we can get a yield penalty. I want to tell you some of our most recent work on understanding how the plant monitors and controls the amount of lignin it deposits, and what we've found was a very unanticipated mechanism that the plant uses to control and link both metabolism and growth with regard to this aspect of cell wall deposition. And then finally, if I have time, I'll conclude by talking a little bit about opportunities for identifying new genes that we can use for engineering lignin to improve biofuel production. So this is some sort of an imaginary, if you will, representation of what the lignin polymer looks like. It's not a repeating structure, so I can't draw you a picture and say this is really what the chemical structure is. But suffice it to say, it contains many different linkages by which these building blocks are linked together in ways that make that polymer very difficult to degrade. That's good for plants because it makes it difficult for fungi to degrade their cell wall, for example, or bacteria. It makes it difficult for us because we might like to degrade it to get access to the polysaccharides in that plant cell wall in order to produce cellulosic biofuels. With regard to plants, it's a very large commitment of fixed carbon, and so it's probably not a big surprise that there are extensive regulatory mechanisms in place that make sure that the plant makes enough and not too much lignin. Perhaps something in the order of 20 to 25% of photosynthate goes into this polymer in plants. As I've mentioned already, it has an important impact on bioenergy and bioprocessing because it is intimately interwoven in the plant cell wall with the cellulose and other polysaccharides that we would like to get access to for biofuel production. And then on the flip side, and another reason perhaps for it being important to understand how the plant regulates lignin biosynthesis, is that it has a very high energy density. So another approach that is being considered is trying to get more carbon into this pathway because of this high energy density of lignin as opposed to the polysaccharides. So why is this polymer important to plants? Well, it's important, the way I tend to describe it is it's important both at the macroscopic level and the microscopic level. So at the macroscopic level, the reason that these trees are the ones out in the garden here can stand upright is that they have this polymer integrated into their cell walls. At the microscopic level, though, lignin is important for rigidifying individual cells. So if I were to give you each a chocolate milkshake and a very thin-walled paper straw, it would come as no surprise to you that that straw would collapse when you tried to drink that milkshake. Well, in plants, including this plant here, oh no, that's not real. It's important to realize that water is not pushed up from the roots, but it's pulled up from the ground by the leaves. So that water is under tension in these cells. Yes, these big open cells here are the ones through which water is transported. And in a plant, this is a cross-section, excuse me, of an Arabidopsis mutant that we work with. In this plant, where these cells have less lignin in them because of a block in the lignin biosynthetic pathway, these cells have collapsed because of that tension, just like that paper straw would. Important at the microscopic level and at the macroscopic level. So why are we interested? Why is there so much interest in modifying lignin? Well, an important, financially significant portion of the effort or the cost that goes into producing cellulosic biofuels is associated with processes like pretreatment. And the idea of pretreatment is to open up, expand the cell wall, disaggregate lignin from the polysaccharide components in the cell wall, so that the hydrolytic enzymes that are used to release fermentable sugars from the wall, those enzymes can have access to those components. And so in this diagram here, the idea is that there's a certain amount of pretreatment that you can do that will give you a certain amount of sugar that you could generate a certain amount of biofuel from. If, on the other hand, you could, for example, decrease the amount of lignin in that cell wall, so the purple bars here are thinner, standing for less lignin, you might be able to use less pretreatment and save money in that processing to get the same amount of sugars out and the same amount of fuel. Or if you found a process of pretreatment that was sufficiently inexpensive, perhaps you could have a more thorough pretreatment, blow that cell wall apart, and get even higher yields of both fermentable sugars and fuels. So the problem with that is that lignin is important to plants for the reasons I've already mentioned to you. So here's the few images of our experimental system, Arabidopsis. We use Arabidopsis because it is a plant we can make very rapid experimental progress with, and the lessons that we learn with that plant can readily be transferred to actual crop plants. Our work would be much slower if we were working with something like switchgrass. At any rate, this is what's called an allelic series of mutants, and these plants have less and less and less lignin in them, in their cell walls, and what you can see is there is a definite impact on the yield of these plants. And that's perhaps not too surprising. I've already told you that water transport is very important, that lignin is very important to water transport, and so if you're interfering with that important process in plants, perhaps it shouldn't be surprising that the plants don't grow as well. And so excuse me, let me just go back and say this is where the focus of our work is, this idea of lignin modification-induced dwarfing. So we see this, in general, we see this phenomenon if we reduce the amount of lignin in plants. There are other examples that I'll show you today where we can change the quality of the lignin without having these severe effects. But this is really the topic that we're interested in investigating, and in fact, Chris McClellan from our GCEP collaborator, Claire Halpin's lab, will be speaking later today about this whole topic again, and I think perhaps might even be mentioning his research with this particular mutant of Arabidopsis. So, I've taught undergraduates biochemistry not long enough to know that few people are interested in actually looking at chemical structure, so I have reduced over the years my biochemical pathway to this sort of schematic. And suffice it to say that this lignin biosynthetic pathway starts with phenylalanine in the top left and generates the precursors of two types of lignin that we call G and S lignin in the bottom right. And the transformations then are shown up here, where the amino acid is hydroxylated and methylated on the ring, and the side chain is reduced to this alcohol, so that we end up with two flavors of monomers that comprise the major components in most flowering plant lignin. And so we have, in general, these are copolymers of these coniferal alcohol and synapyl alcohol, so-called G and S lignins, respectively. So that's the biochemical pathway, but we need to consider this in the context of, in a cellular context. And so all of those biochemical processes go on within the cell at the top left here. These monomers are then excreted to the cell wall, where they're activated by peroxidases and lactases to form their free radicals, which then will spontaneously couple to generate this polymer in the wall. Now, the reason that lignin manipulation is such an opportunity is that it's a very unusual biochemical polymer. For those of you who want to think back to your biology classes, you may remember that nucleic acids like DNA and RNA, those are synthesized. So DNA is synthesized on a template of DNA. RNA is synthesized on a template of DNA. Protein is synthesized on a template of RNA. We're using that information. Polysaccharides are a bit different. Their structure is very carefully dictated by the enzymes that assemble them. But lignin is entirely different. Lignin is a random radical coupling process where what the lignin polymer that you get out is dependent solely on the various monomers that go in, that the plant produces. So if we go back to this slide, each of these three, this paracuminal alcohol is a very minor component of most plant lignin. These are the two major ones, as I introduced before. So the ratio of these monomers synthesized determines the nature of the polymer produced. So we have a few pieces of experimental evidence that backs that up. So for example, one of the things that's really fun that we can do in Arabidopsis is that we work with mutants that are blocked in these biochemical pathways. I showed you one before, those various dwarfed mutants. But we identified a mutant that was blocked at this one step called F5H, which is required for the synthesis of syringil lignin. And if you knock that gene out, what you get is a lignin that's produced only from these G subunits. These plants, as I'll show you in a minute, grow perfectly fine. Perhaps not surprising because that pine tree out there also makes lignin that's only derived from G subunits. So we know that's a functional polymer. If we instead overexpress this particular gene, what we find is that we can reroute carbon just to these S subunits and make a polymer derived only from S subunits. And as I said, these plants, this is just a photo of normal plants and the plants that make only the G lignin. The S lignin synthesizing plants look just the same. These plants tolerate this change in lignin very well. And in fact, we've taken that technology and transferred it to poplar, which is a bona fide bioenergy crop. And the easiest way for me to show you what the impact of that biochemical change in lignin is to show you these samples of paper made from these modified lignin poplars. The wild type plants here with normal types of lignin, you can see this brown coloration is because of the residual lignin left in this paper. That's why paper towels are brown in color. But under the exact same conditions, the high S lignin poplar wood is white because all of the lignin can be removed. So there's a very distinct impact of this change in lignin chemistry on our ability to remove it. And that's where, although this example comes from pulp and paper, we can expect similar impacts with regard to biofuel production. Okay, so those were the good stories. The interesting thing, or an interesting thing, came when we looked at mutants that were blocked earlier in this pathway. If you block C3'H, again, we identified a mutant that was blocked here. What we found was that these plants started making lignin from this subunit that I told you was a very, very small component of wild-type lignin, less than 1%. But the narrow line I've shown here is meant to represent that the the flux to lignin, the total amount of lignin synthesized, was dramatically reduced. And the impact on growth was apparent. Okay, so I don't even know if you can see this little thing here, but that's the mutant that is blocked at C3'H. So there's a very significant impact on yield. And if you talk to a plant breeder, they will tell you there are 3 things that are important in breeding crops: yield, yield, and yield. So this is a very important thing for us to get a handle on and to understand why this particular change would lead to such a dramatic phenotype. I would say, though, also that, I won't show you the data, but these plants also, as I said, have lower levels of lignin. They have collapsed xylem, the paper straw issue again. And so perhaps, again, it's not a big surprise that these plants don't grow well. They can't transport water properly. So this is where this particular story, if you will, collided with another piece of research going on in my lab. One of the interesting things about taking a genetic approach to understanding plant biology is that instead of being theory-driven, as many of the talks that we've heard over the last 2 days are, when you use genetics, you let the organism tell you what's important. So we identified another mutant that had lower levels of lignin, like some of the ones I've already told you about today, and that also showed this phenotype where this low level of lignin in the water-conducting cells led to a collapsed vasculature. We didn't know what this particular mutant was, of course, when we first identified it, but we have technologies for going from a mutant phenotype to a gene. And we succeeded in doing so. And we found that because the Arabidopsis genome has been fully sequenced, we can move this sort of science along quite quickly. We found that this gene that we called REF4 had a paralog, another gene quite like it, that we called REF4-related 1, or RFR1. And what we found was that when we knocked those genes out, so this is a REF4 knockout, this is an RFR1 knockout, this is a plant in which we've combined both of these knockouts into the same plant. Those plants didn't look the same as this initial mutant that we identified. This is this mutant with low lignin, collapsed xylem, and again, it's dwarf. These ones are not. But when we did some biochemical analyses of these plants, and this is using, oh boy, did that axis ever get messed up. Never mind. This is looking at a soluble metabolite that's derived from the same pathway as lignin. We found that our original mutant had lower levels compared to a wild-type plant, but when we knocked out those genes, the accumulation of this compound went very much higher. So, if you will, what we have, if I try and make a traffic light analogy, is that in these plants, this metabolic pathway leading to that soluble metabolite I told you is being properly regulated. Not too much, not too little, but it's under active regulation. When we knock these genes out, then the pathway is elevated in expression and activity, and we get higher levels. The plant is moving forward with that biochemistry to a greater extent. And when we have this particular mutant, the mutation in this particular gene has put a constitutive brake on the pathway. And so there's clearly something that's very important about this protein that acts in a regulatory role in this biochemical pathway. So what does this gene encode? It turned out that this gene encodes a component of what's called the mediator complex. The mediator complex is one that binds upstream of genes. So in this particular diagram, we have a gene illustrated down here. This is the mediator complex. It's about 30 proteins that get together to interact with proteins that called transcription factors that regulate the expression of genes. So you can imagine, and it's the case, that there are proteins that bind upstream of genes that tell the cellular apparatus how much of a particular gene to make, under what stimuli, abiotic stress or biotic stress, and in what cell types. And what this mediator complex does is that it integrates all of that information to come up with some sort of a concerted output of gene expression. And apparently this particular mediator component, which is this one called MED5, is important in regulating this lignin biosynthetic pathway. So, as I said, some of our work sort of collided in this project. And so one of my postdocs asked the question, well, if this mutant that is blocked early in the pathway that is so dwarf is somehow perturbed in the regulation of lignin synthesis, and this protein called REF4, or we now call it MED5 now that we know what it does, is an important regulator of the pathway, is there any interaction between these? Can we test that in vivo? And so what he did was generated a mutant where both of these mediator components were knocked out and the biochemical pathway was also blocked. And so instead of being this very sad little dwarf plant, this plant can now grow. This plant is still blocked in the biochemical pathway leading to lignin synthesis, but it is no longer dwarfed in this way. And that was a totally unexpected finding. What he found was that in these plants, lignin biosynthesis was restored. So here's the low levels of lignin in the lignin biosynthetic mutant, but if you eliminate these mediator components, the lignin goes up again. The cell wall defects that we saw that associated with the collapsed xylem, so here are some TEM images of, here's the plant that has low levels of lignin and the wall is very distorted. It's restored basically to normal when you knock out these mediator components. And in work done with our GCEP collaborator, John Ralph's lab, they took a look at the lignin and found that it has an entirely different structure. It has a structure that reflects the sole contribution of these para-hydroxyphenyl units, the units that I said this plant would now make as a result of the metabolic block. What's fascinating about that is that nobody thought that this type of lignin could even be made by plants and that it would be functional. But now we have plants that make it just fine. So this is a 2D NMR basically looking at the contribution of S, G, and H units to the lignins of wild-type plants, MED5 mutants, and this very unusual triple mutant. So in the last couple of minutes, I'll finish up just by telling you some insights that we got from doing experiments that are called RNA-seq experiments. So I'm sure you all know that there have been tremendous advances in DNA sequencing technologies in the last decade or so. Not only can we sequence DNA and get the sequence of the human genome, we can sequence the RNA made by any individual organism. So instead of telling us all of the genes in an organism, this type of experiment tells us what genes are being expressed at any particular time. So we could ask what genes are expressed as a result of UV or insect attack or mineral deficits. Here we ask the question, what's the difference in gene expression between these mutants that we have that are either blocked in mediator, blocked in lignin synthesis, or both? And so the image on the left is the genes Venn diagram for genes that are upregulated. On the right, those that are downregulated. And what you see is that there's a huge number of genes that are upregulated and a huge number of genes that are downregulated because of this metabolic block in these original mutants. And perhaps probably the most important set of data is when we combine this mediator knockout in the same genetic background, this number 4,400 goes down to a few hundred, 3,300 goes down to 200. So whatever the responses are that lead to these changes in gene expression in this lignin mutant, they are mitigated if you knock out mediator. So our model is that, again, a model of a process that we didn't even understand existed in plants, is that when we block this biochemical pathway of lignin synthesis in the cytoplasm, changes either in cell wall chemistry or structure or soluble metabolites, and that's actually the model I prefer, are somehow detected by a sensor. This signal is transduced to the nucleus where mediator is required for changes in gene expression, and this set of changes in gene expression then have a trickle-down effect. They cause a cascade of other changes in gene expression that result in the developmental defects we see in the plant. I think the best way to think about this is to think about anaphylaxis. We know that our immune system is here to keep us healthy, but in certain individuals, when they, for example, if they have a peanut allergy, their immune system can go haywire and make them very, very ill. Here, this system is involved in keeping that phenylpropanoid gene expression at the right level, but if we have a block, we can have very severe changes in gene expression that actually end up making the plant sick. So I'm running out of time, so I'll just say that another one of our GCEP collaborators is studying what other genes may be required in this sensing pathway and has identified other mutants in which this dwarf phenotype is suppressed. These plants can now grow quite healthy, and he's in the midst of cloning these genes. And I think I better just go ahead and skip some of these data and just finish up. Some of that was on Whitney's poster yesterday, and just say that what we found is that a previously unknown regulatory circuit that keeps lignin biosynthesis at an appropriate level, and that this involves this mediator complex. We found that we can reverse this dwarfing that we see in low lignin plants, and it's actually not simply an architectural problem, not simply caused by defects in water transport, xylem collapse. It's an active protein mediator process. We found that we can make a whole new type of lignin that could be used in a biorefinery, and that we have an active set of feedback mechanisms and that are detecting phenylpropanoid metabolism and cell wall changes, and that's really the focus of our current GCEP work. And with that, I'll just say thank you to those people who are involved, both at Purdue and our DOE-funded EFRC called C3 Bio, as well as my GCEP collaborators, and I'd be happy to take any questions.
Okay, do we have any questions for the speaker? Sally.
I'm not a biologist, so this is a non-biologist question. So this is really fascinating, and it seems like you've made such incredible progress. When and how would this knowledge be, moved to the next level where you could start incorporating it in, actually biofuel crops, and what's involved? Is that 50 years from now? Is that 5 years from now?
Oh, okay.
Yeah.
Yeah, it's a great question. I mean, I have one supplementary slide because I really wanted to keep the focus of this talk on the fact that that we've discovered a novel regulatory mechanism. It's too blunt an instrument to deploy in the field because this mediator complex is involved in regulation of many, many biochemical processes within the cell. And so, for example, we already know that these plants are more sensitive to pathogens. But what we have done is a simple experiment to ask, does this change in lignin, now we have plants that make pure H lignin, is there any direct application for that? And what we found is that even if you don't do pretreatment, the dotted line on the left shows that we can release more sugars from the cell wall with this novel lignin than if we have the typical GS lignin of wild type plants. So that says, we're making a step in the right direction. And so what becomes really important is that we study this system, identify some of the other players, and identify those tools that will allow us to get a better handle on controlling lignin synthesis and this dwarfing without leading to these pleiotropic phenotypes like pathogen susceptibility. So, you know, it used to be that not so long ago, cloning one of those genes was the work of, that was a PhD in biology. Whitney cloned 15 of them in a month not too long ago, and I had to skip over some of that data for the sake of time. So the rate at which we can bring these genes, this knowledge to bear is very much reduced, and so that's where the exciting things come in, is how can we take this information, integrate it into things like breeding schemes to really improve biofuel crops.
I have kind of an off-the-wall question.
Well, it was an off-the-wall presentation, so.
It was really interesting. So if you drop an atmosphere, you get about 10 meters of height with water. And so you say that, you know, the height of plants is limited by their ability to withstand the strength of being able to pull the water up. But yet trees go to 10 times that height, roughly, with some other mechanism. Have you thought about trying to incorporate those other mechanisms to combat the dwarfism associated with weak cell walls?
Okay. So those weak cell walls are collapsing under water potentials that are very, very low. I mean, a Arabidopsis only gets this big. So that issue of how far can water move up a capillary in this country, it doesn't come into play. Lignin is important for not just that structural support, but the making the vasculature more hydrophobic so that you don't get adhesion of water to the very hydrophilic polysaccharides in the cell wall. I don't know that, we don't know to what extent that is tied into limitations on how tall plants can grow.
Hi, Clint. Hi, this is kind of a random question as well. There's quite a few examples of knockouts in genes that give different types of lignin, less lignin, H-lignin, and examples where sugar is released more easily. So do you have a feel for, at this stage, what you would like to see in a crop? Like what modifications you think would work in terms of a biofuel crop? What would be optimal? Do you think it would be one modification or there'd be several? And this would be something you might learn over the next few years?
Yes. So we know, for example, that if you block the lignin biosynthetic pathway very early, such that the plant simply cannot make it no matter what you do. These mediator knockouts do not rescue that phenotype. So at some level, lignin is required. I think what's exciting about this is that this research gives us the opportunity to revisit technologies that had been discovered and abandoned because of the deleterious phenotypes that were associated with them. We're now going back in the lab and revisiting those and asking how many of those dwarf low lignin plants or altered lignin plants, how many of them can be rescued with this type of an approach so that we can think about taking those abandoned technologies off the shelf and incorporating them into breeding programs.
Question about the, actually you've displayed a very interesting thing about evolution, perhaps, exposed why there are all these different pathways and what has actually been selected over the millennia to be the one that's preferred by the particular plant, right? So, but my fundamental question is, we learned from the corn ethanol problem that caused a lot of malnutrition around the world, that getting 7% of sunlight to produce sugars and then spending energy to extract and then ferment and then mix with a little gasoline to denature it and burn it at less than 1% net efficiency is really not the way to go. So what do you think overall this says about biofuel idea, which I don't feel makes a heck of a lot of sense, but.
Yeah.
but-
So I think there's certainly many varying opinions on that issue. I guess I would argue that it's too early to abandon any type of technology. We have been, if you think about corn, we have been breeding corn for 5,000 years for grain characteristics. And still, over the last 80, 90 years, the theoretical grain yield has been increasing steadily. So there's a huge opportunity there. We have been breeding plants for their cell wall characteristics for essentially 0 time. And so, at the end of the day, it may be the case that what we really need to do is improve the caloric value of the plant cell wall by increasing lignin synthesis. Important to understand how it's regulated. Combust it, do something with it, sequester that carbon. The fact is that as I understand it, there is no technology that competes for recapturing CO2 from the air, which is a problem that we're going to have to tackle, that competes with photosynthesis. So at least for the time being, that's a way to think about it. But whether biological conversion like fermentation or catalytic conversion is another thing that we're exploring. How those will turn out, it remains to be seen. But I think it's still an area worth investing in.
Hey, our time is up for now, but I'm sure the speaker will stay around to answer questions during the break. Let's thank him one more time.